Welcome to Bio 212, Anatomy and Physiology 2. And what we're doing now is we're moving to Chapter 21, which is the structure and function of blood vessels inside the human body. This is the focus of our conversation today. And I just want to dive right into this because, for the average adult, you're looking somewhere between 90 and 100,000 miles of blood vessels. And those are arteries, capillaries, venules, veins. And for the purpose of our discussion, the significance of the capillary bed can't be understated, okay? Or overstated. I'm not quite sure what the best phraseology is for that, but the focus of your studying when you're reading the book before watching this video is that you understand clearly that the whole purpose of these blood vessels is to get materials to the capillary bed so we can perfuse tissues, right? Having said that, the value, because I was looking up the value when I started, right before I started this, it doesn't include lymphatic structures. So you're going to throw in a couple, 1,000 more miles, probably like 24, something like that. It's a one-way structure. Maybe 30,000 miles of vasculature for the lymphatic system as well. But how do we actually build them? Here is the image inside of Tortora and Derrickson. And you can see here's the lumen of the artery and the lumen of the vein. And you can tell here that blood flow in this direction is going to move such that if blood moves through a vein and the pressure drops, it stops blood from moving backwards because here are the valves that are going to stop it. But more importantly than, understanding that, hey, blood's going to move through arteries and veins to get to a capillary bed, let's think about how Mother Nature designed these, okay? Because there are layers that you want to make sure you understand. These are layers or tunics. That most internal part here is also called the intima, and that is the endothelium that sits on top of a basement membrane. Likewise, inside of a vein, you can see endothelium sitting on top of a basement membrane. You have to see that inside a capillary bed as well. The media is going to be different depending on the types of the 5 vessels you're looking at. The larger the artery and the vein, the more smooth muscle and supporting structures you have to have inside of it for it to do its job. But at the end of the day, see if I can figure out a way to do this. Cool, let's go like this. Let's go like this. We're going to have something that comes. This is blood coming from the heart to a capillary bed. Doesn't matter which capillary bed it is. It has to now perfuse the tissues, which means we have to build something that's going to allow for movement of materials. Okay? And the idea is this: rich oxygenation and glucose, perfusion of tissues at capillary bed, collection of waste materials to return back to the right atria. So this is going to be our left ventricle. Let's go right atria over here, RA. And this is going to be, go back to red. Oops, got rid of it. Anyway, the red would have been left ventricle. Okay? So that's our introduction. And what I mean by that is, look, you have some, if we go from the outside, you have the tunica externa. Okay? Where are you? Tunica externa, way out here, right? And then you actually have some smooth muscle, and you actually have some elastic lamina. Inside the media, you actually have some muscle and some lamina as well as a basement membrane for that endothelium. So that's like the third time I've stressed that to you. Make sure you understand why Mother Nature has built these. And you're going to notice you're never going to find a valve inside of an artery, but you will find valves inside the lymphatic system. Okay. There are 3 types of arteries, okay, there. The idea, of course, is they actually have to have a specific ratio of muscular to elastic tunica, right, because we have to withstand the force that's coming out of the left ventricle. It has to have high compliance. As you age, obviously, that compliance goes down, but it should stretch and expand in response to the pressure without tearing and all the red blood cells that are pounding around inside of it. You should always keep inside the consequence of vasoconstriction. Yes, it's going to decrease the lumen diameter. Yay, you got that. Woohoo! What happens at the capillary bed? Okay, is the pressure too high? Is the pressure too low? Vasoconstriction is supposed to maintain the optimal diffusion was going to draw another one of those capillary beds for you here, so let's just do it quickly. We already have blue, so let's go like that. There's that right there. Because you want to be thinking about Mother Nature, because she doesn't really care that the blood vessels are there. I mean, they're important, don't get me wrong, but they serve a purpose, and that purpose is to get materials to here, right? This is the capillary bed. This is where things are actually happening. Yeah, you got to keep the capillary, I mean, the arteries, arterioles, diffusion, collection, move materials out. That's all important, don't get me wrong. What I want to make sure you're always thinking about, though, is what can change this, right? And vasoconstriction decreases the space here. Well, what happens to the pressure here? Likewise, vasodilation happens here. What happens to the pressure here? Okay. We only spend maybe 30 seconds thinking about elastic arteries, and the major elastic artery, of course, is the aorta, and that aorta has within it a lamina and elastin, and what's going to happen, of course, is as blood moves into that space, it pushes these things out, and that's really what establishes the pressure inside the circulatory system. So if you have less volume moving into this, well, you'd expect less pressure inside the system. That's exactly what you see. To compensate for that, your nervous system will detect this and increase vasoconstriction of blood vessels, though. So what is an artery? Well, you have different types of arteries. You have muscular arteries. They have relatively thick walls. They're capable of vasoconstriction and vasodilation, and they are also called distributing arteries. These are the 25 or so arteries you guys have to know inside the body to get blood to your toes or your arms or your hands or something like that. What Mother Nature can do, of course, is she can create collateral circulation by making these things that are called anastomoses. And we saw that inside the heart. We haven't really looked for it anywhere else inside the body, and we probably won't either. Arterioles, these are abundant microscopic vessels, and where I need you to pay attention inside the figure in a couple of minutes is here. Whoops, that's not what I wanted to do. Here's a meta-arteriole that has a pre-capillary sphincter. So a specific location before the capillary has a sphincter, which monitors blood flow into the capillary. And sympathetic innervation and local chemical mediators can alter the diameter of that. So think nitric oxide, think epinephrine, think norepinephrine, right? And what's going to happen is by doing that, you change the flow rate. Even though your textbook says there are things that are such as resistance vessels, right? I think I would caution you to think about every blood vessel actually being a resistance vessel. Every time a red blood cell hits the side of the wall of the lumen of a vessel, it slows things down. So that means the farther the distance, the more resistance you actually should encounter inside your circulatory system. And resistance is the opposition to blood flow. Okay, so even though we're talking about what's happening at the capillary bed, it's an arduous route to get there. Finally, capillaries themselves, okay, they lack tunica media and tunica externa. However, they usually arise from a single meta-arteriole, which means it actually has vasomotor and through-fare channel responsibilities, right? So vasomotor meaning it can have intermittent contraction and relaxation depending upon the needs of that tissue, but also the needs of the entire body. Likewise, if you just need to get the blood out of there, meaning through it, you actually have one through-fare channel for each capillary bed, and you can see that here. So here's blood moving through the heart. There's that meta-arteriole we were talking about, and here are the precapillary sphincters, right? So Mother Nature needs to stop blood flow into these branches off to the side. She collapses all of these, and now blood can just travel right through that through-fare channel and get back to the muscular venule and then travel back to the heart. Okay? That's actually a fairly significant idea, which means that Mother Nature can stop blood flow when she thinks it's important to essentially any part inside your body. And we have three different types of capillary beds. The ones we've been speaking about so far are the continuous ones. These are endothelial cell membranes, okay, and they have tight junctions, and they have a basement membrane that's made up of collagen, and if you rip this apart, you activate the clotting pathways that require the fragments of megakaryocytes that you know as platelets. Okay, we also have fenestrated though capillary beds, right? And those fenestrated capillary beds are going to be located in specific parts of the body. You'll read about them inside your textbook, but you know what's going to happen here. We can push slightly more volume through this because there are holes that are traveling through it. And then finally, you have these sinusoidal structures. These are located usually in areas where blood pressure can be maintained easily. You'll notice that they have very little connective tissue holding them together, and there are spaces in between them. This allows for egress. So think about how cells get into and out of the circulatory system, say inside your bone marrow and stuff like that. We're thinking about a portal vein, okay, the hepatic or hypophyseal, or if you think about fetal circulatory structures, blood passes through a second capillary bed. And when we were talking about the hypophyseal portal system, I tended to stay away from this idea because what I want you to understand is that Mother Nature needs to slow blood flow down so it can collect materials or deposit those materials. I don't tend to really think of it as a second capillary bed, but your textbook describes it that way, at least inside chapter 21. You'll notice it didn't describe that way inside chapter 18. Okay? Now, as we're moving back towards the heart, towards veins, what I want you to do is see that you actually have the counterpart to arterial structures. So that means, generally speaking, once you know the arterial names, you know vein names, okay? And these can actually create exchange units with capillary beds themselves. And the farther you go towards the heart, the more muscular they actually become. Veins themselves, structural changes are definitely not as distinct as they are inside arteries. They have those same 3 layers, and they also have those valves to prevent backflow. Because remember, when we actually get towards the heart, let's see what color comes up here first. It's probably going to be red, right? Yeah, of course. So let's go like this. Wham, wham, wham. Okay, and now we're going to go blue. The next piece of information I'm going to be layering in on top of this now. How many times have I drawn a capillary bed for you? All right, blood flow is now returning back to the heart. The pressure here has to equal about 0 millimeters of pressure. Okay, and you'll see it's going to start out somewhere around 100 over here. Okay, so how does this all happen? And why does Mother Nature do this? Well, because believe it or not, she actually looks at the veins in the body as though they are a blood reservoir. Your pulmonary vessels get 9% of the blood, your heart gets 7%. Systemic capillary beds at any given point in time are rocking a whole 7% of all of the blood. So all of this work is being done for 7% of it, which means all of the materials that need to be stored and have access to. Remember, we're talking about you have 4 to 5 times the ability to play with the cardiac output. Well, guess what? This is where it's going to come from. Okay, because we can only get 7% of it inside that space at any given point in time. And as you increase the activity, the percentage of used systemic capillary beds even gets smaller. Okay, so capillary exchange. There are going to be 3 types of capillary exchange or methods that are used. Diffusion, transcytosis, and bulk flow, where diffusion is substances move down their concentration gradient. They cross past the capillary wall through intracellular cliffs and fenestrations. And most plasma proteins cannot cross. That is really important because that's actually how we drive osmolarity both outside of the capillary bed and inside the capillary bed. Okay? There is a blood-brain barrier where the tight junctions limit diffusion, but obviously oxygen gets through and obviously glucose gets through. It's the other stuff you want to keep out. Transcytosis is where you literally pinocytose it up into vesicles that enter the endothelial cells and then exit by exocytosis on the other side. And these are for large lipid-soluble molecules. Okay? So depending on where we're at, if you think about, say, the ventricles inside the brain, this would be one way to make sure you get materials through. Bulk flow, of course, is the passive movement where large numbers of ions or particles and fluid move together in the same direction. It's based on the pressure gradient as things are moving through the capillary bed. The arterial side, bulk movement outward is easy. Reabsorption on the opposite side is what's actually going to happen. That's actually what we're going to call this. Bulk flow drives on the capillary bed side what we call filtration. it also drives reabsorption on, say, venous blood return side of a capillary bed. Okay, so think those two sets of triangles with a capillary bed in them, right? And the capillary bed will actually have opposites. So this looks formidable, but it's not. It's blood hydrostatic pressure. So let's draw in red here. Blood, oops, let's do blue first then. Okay, look, here's the capillary bed. Now switch over to red. Wham. And this is like this over here, right? So you have a certain amount of pressure that's pushing everything into this capillary bed. That is the blood hydrostatic pressure. It's pushing materials out. Okay? As a result of that, you have interstitial fluid osmotic pressure, okay, meaning around the spaces here. And what's going to happen is we're going to subtract that by the blood colloid osmotic pressure. Remember I said a moment ago that if we leave proteins behind inside of the capillary walls, it's going to pull fluid back into this. Okay? And that's actually, switching colors here, go back to blue, part of the forces that drive the movement of materials back into the oxygen return portion of the capillary bed. Okay? And you also have interstitial fluid hydrostatic pressure. You push all this fluid in this space, it needs to go back where it came from because there's only so much room. There's all these cells in here and all that sort of stuff. So sort of get a mental image of fluid dancing around cells, and if the pressure gets to be too great, you got to push it right back in here. There are two pressures that promote filtration though, okay? So I'm probably working with blue here. Blood hydrostatic pressure actually is generated by the pumping action of the heart, and it's going to fall to about 16 millimeters as it's going through the capillary bed. It starts at around 35 millimeters here, it gets to about 16 millimeters here. And the interstitial fluid osmotic pressure is also driving at 1 millimeter of pressure. And a millimeter may seem like that great of a number, but without it, the system doesn't work. It's actually awesome engineering the way that the osmotic pressure pulls and pushes things around your body. Okay, filtration, to finish this off, you have blood colloid osmotic pressure as well as interstitial fluid. These are the values over here, right? And I've already explained them to you, so I'm not going to draw the cartoon again, but notice what we're talking about. We're reabsorbing materials now back into the capillary bed, and you'll see the cartoon of it in a minute. Let's go like that. Together, this is Starling's law, where nearly as much is reabsorbed as was filtered, but obviously the gas goes to the mitochondria, the glucose goes inside the cytosol of the cell, and we pick up the carbon dioxide. Okay? At the end of venous, you'll see the equation in a second, you have to have a negative value as a sum from the equation. That negative value means that we have correctly delivered materials to the cells that that capillary bed supported. Okay? So what happens to any excess fluid inside that space? Well, here's blood flow traveling in, 35 millimeters per blood hydrostatic pressure. Okay? Blood colloid osmotic pressure is 26 millimeters. So you're going to do the math down here. 35 plus 1, there's the 1 right there. It's 26 minus 0, which is, oh, where's the 0? Right there. Okay? And now what you do is, this is your net filtration to give you 10 millimeters on this end. 10 millimeters on this end to filtrate materials out. And now you're going to reabsorb materials. So you're now going to push materials back in here. Okay? And that's actually how a capillary bed works. Excess fluid ends up going inside here, though, the lymphatic system, where it's then delivered to the rest of the body. Okay, so this is a pretty good place to pause, right? I want to introduce one idea, though. Factors that are going to affect that blood flow, okay? Total cardiac blood flow, okay, is cardiac output, and distribution of cardiac output depends on the pressure differences that drive through these tissues. And I know I've stressed this again and again and again. Just because your heart rate's going up doesn't mean you have more efficient net filtration and net reabsorption pressures inside a capillary bed. And that's where we'll pick up.